Hi class, today I will be sharing some information on Vibrio, a genus of bacteria. I first became familiar with Vibrio in the summer of 2016 when my husband was sick with flu-like symptoms. After several days of severe gastrointestinal upset, abdominal cramping, and intermittent fevers, we took him to the emergency room for evaluation. After some testing, his results came back positive for vibriosis, a condition resulting from exposure to pathogenic Vibrio species. I wanted to take advantage of this opportunity to learn more about the microbe that I had interacted with, but understood very little about. Members of the Vibrio genus are prokaryotes and belong to the domain of bacteria. Furthermore, they are grouped into the phylum of Proteobacteria, the class Gamma Proteobacteria, the order Vibionales, the family Vibrionaceae, and the genus Vibrio. We'll focus a little bit more on the characteristics of the genus before moving on to specific pathogenic species. Members of the Vibrio genus have distinct morphological and metabolic features. Their name, in fact, refers to their Vibrio shape, which is a curved rod and is often described as a comma shape. They are gram-negative, non-endospore forming, and interestingly enough, can alternate between a monotrichous and a peritrichous flagellar arrangement, depending on environmental conditions and the motility needs of the bacteria. Their diameter is approximately 0.5 micrometers, while their length varies from 1.5 to 3 micrometers. They can be observed singularly or in arrangements of long S-shaped chains. Metabolism and growth requirements vary between various Vibrio species. V. cholera, for example, is a halotolerant organism that is known to be transmitted via freshwater systems. Most Vibrios, though, including V. vulnificus and V. parahemolyticus, are actually halophiles, meaning they not only tolerate saline environments but require them. Both of these organisms thrive in the salty concentrations of oceans and estuaries, and can also be present in the brackish water of river mouths, where freshwater and saltwater mix. Optimal growth occurs at 2 to 2.5% salinity. They prefer a slightly basic pH between 7 and 9, a temperature of approximately 25 degrees Celsius, and they are facultative anaerobes, preferring the metabolic processes of cellular respiration, but capable of surviving low oxygen environments via fermentation of glucose molecules. The most well-documented pathogenic species of Vibrios are V. cholera, V. vulnificus, and V. parahemolyticus. While all three of these pathogens are concerning and worthy of note, we'll be focused on V. parahemolyticus, which is currently reported to be the most prolific of the three. According to the CDC, V. parahemolyticus is the leading cause of seafood-related foodborne illness in the United States, with up to 80,000 cases per year. The majority of these are transmitted through contaminated foods, typically raw or undercooked shellfish. Other modes of transmission include exposure to the bacteria at an open wound site, which is common for fishermen and individuals working and living in coastal communities. V. parahemolyticus was first discovered during a severe outbreak of gastroenteritis in Osaka, Japan in 1950. Utilizing Robert Koch's four postulates, V. parahemolyticus was isolated, cultured, and eventually found to be the causative microbe. In 1971, the first case of acute gastroenteritis caused by the species was reported in the United States, and since then it has been documented across the globe, with major outbreaks reported in North and South America, Europe, India, and across Asia. Signs and symptoms of V. parahemolyticus infection vary depending on the method of entry. Gastrointestinal infections can result in fever, chills, abdominal cramping, watery diarrhea, and nausea vomiting. Wound infections can cause fever, redness, pain, swelling, and warmth, as well as oozing at the wound site. Several factors are thought to contribute to the pathogenesis and virulence of V. parahemolyticus. Virulence in many cases starts with the pathogen's ability to adhere to host cells to initiate infection. V. parahemolyticus expresses the gene for MAM7, a protein that is characteristic of the outer membrane of some gram-negative bacteria. The MAM7 molecule can form attachments to two components in the cell membranes of human host cells, the protein fibronectin and the lipid phosphatidic acid. Additional virulence factors include exotoxins. The two most widely discussed and researched include thermostable direct chemolysin, or TDH, as well as TDH-related hemolysin, or TRH. Both TDH and TRH are hemolysins and can disrupt the cell membrane of red blood cells, leading to cell lysis. Research suggests that TRH and TDH allow V. parahemolyticus cells to access and utilize valuable iron stores that are protein bound within the red blood cells of human hosts. V. parahemolyticus provides us with an example that highlights the role of bacteria in foodborne illnesses. Through our study of Vibrios, we can observe bacterial mechanisms that increase virulence and thus increase a pathogen's chance of survival and proliferation within the environment and within a host. An understanding of how microbes interact with their environment under various conditions is crucial for management and mitigation of health risks, especially as we navigate climate change and evolving food systems. Avoiding V. parahemolyticus is possible. If you are a fan of seafood and shellfish, opt for cooked variations. The CDC website provides recommendations for boiling, frying, steaming, and baking shellfish to the appropriate internal temperatures. If this is not possible, avoid consuming raw seafood and shellfish between May and September, when warmer waters may encourage larger numbers of bacteria.
Hello, my name is Izzy Murray, and I wrote about the bacteria Treponema pallidum. Just some quick facts about Treponema pallidum. It's a sexually transmitted disease that causes syphilis. It's also a gram-negative bacteria. It's spirochete in shape, meaning it's very motile with important flagella that move it counterclockwise and clockwise. It's microaerophilic, which is important because it can grow without oxygen. It doesn't need a lot, but it does need some to survive. And it also must be in a human host, so it can't live outside of one. I also wanted to bring up the phylogeny of Treponema pallidum. It's kind of an enigma of how it all started, especially because it's increasing in rapid numbers, especially even today. So the first one is the pre-Columbian hypothesis, where it talks about how treponemal diseases in general over time and how they respond differently depending on changes to the environment. So that can mean a lot of things, whether it was temperature or climate. And then the other one is the Columbian hypothesis, which deals with Christopher Columbus and the spread of syphilis. The reason why people think that this is one of the huge parts of the spread was because of what was documented between two physicians who talk about syphilis, and then dating back skeletons from that time period was found to have syphilis-like lesions on them. So yeah, there were these two physicians that talked about syphilis and how it was kind of spreading everywhere and then spread to the US from Spain. Another thing I wanted to talk about was the stages of syphilis. So it's kind of broken down into 4 stages: the primary, secondary, latent, and tertiary. While syphilis is treated with penicillin, when it gets into the last 2 stages, it's kind of unlikely to be resolved and is usually just stays in the body forever. But the primary stage is categorized by enlarged lymph nodes, and then usually a lesion in a genital, oral, or anal region, kind of wherever that contact was transmitted. Usually the lesion is painless. And then the secondary stage is kind of the classic stage for symptoms, or at least one that most people have heard about. It's the rash on the palms of hands or the soles of feet. And then usually this is also associated with a fever. And then the latent stage is the chronic one. It's less likely to be resolved here. This is also a huge part where congenital syphilis can happen, which is syphilis being transmitted to a fetus from a pregnant woman if she has the infection. And then usually at this time, the fetus would be born with syphilis. And then the tertiary stage, the last one, this is the neurological damage one. It also can impact other organs, but it's most known for impacting the neurons function. Another note I wanted to mention was the historical impact of syphilis in medical ethics. So the Tuskegee syphilis study was a 40-year study starting in 1932. It examined 600 African American men in Alabama, and about 400 of those people had active syphilis, while the 200 were control. These men thought that they were being treated for Treponema pallidum, but the researchers were lying to them in order to see the effects of Treponema pallidum in the human host long term. This is, of course, a very horrendous study, and it did become a gateway to protecting human research subjects and ethics for future medical experiments, and it is mentioned in medical ethics courses today. I also wanted to talk about my art component, so I decided to make a puzzle with little spirochetes on it. I made it a puzzle specifically because, like I was mentioning before about the phylogeny, it's kind of hard to know exactly how syphilis spread. There's ideas, but it's kind of an enigma. They're also pink to represent them being gram-negative, and the white background is to envision them under a microscope. I also drew some of them, if you notice and look closely, as little snakes, because today we kind of look at syphilis and spirochetes as like little snakes. They're complicated, they can be wound up and kind of have that corkscrew shape. I don't know if anyone's ever seen the little plushies of bacteria or viruses, and syphilis is represented as a snake. So I thought that would be kind of fun to add that in here. Anyways, thank you for listening.
Hi, my name is Nick Nesselrode, and I'll be walking you through my graphic novel on the genus Streptococcus. So I'll start with a picture of the whole page and then kind of zoom in from there. Streptococcus is a gram-positive bacteria. They are catalase negative, non-motile, non-spore forming. They reproduce via binary fission. They belong to the phylum Firmicutes. There are more than 50 species of Streptococci, and they are facultative anaerobes, most, although some are obligate anaerobes. Some common illnesses that are caused by Streptococcus would be scarlet fever, strep throat, pneumonia, some neonatal infections, which we'll talk about later, and even UTIs. Some of the pathogenic species are classified into groups, and these are based on some classifications done by Rebecca Lancefield, and they're based on their hemolysis and the antigens that are produced by each of the different species, so they can be classified into A, B, C, G, and there's more. This is the cell wall of our gram-positive Streptococcus. So in the middle, you've got the cytoplasm, then the phospholipid bilayer, then a thick peptidoglycan layer of those NAG and NAMs and the cross bridges between. Then you've got some lipoteichoic acids that go through it and some pili that can come off of it. Group A Streptococcus have something called M proteins that aid in attachment and a hyaluronic acid capsule that actually keeps it from being phagocytized by other parts of our immune system. And so this helps it to be more virulent. So you can see Streptococcus divide by binary fission, and they are circular. So you can see in the first image that it is a single strand of DNA, then the DNA replicates and goes to opposite ends, and it starts pulling apart in that divide in the middle. Then it finally splits into two, and oftentimes the streptococci just stay connected together, and in some different species they're diplococci, and so they'll just stay two of them stuck together, and then others are forming long chains, which we know of as the kind of classic streptococcus. We'll go through a couple of species here that are likely most notable. So group A, Streptococcus pyogenes. So this one is the cause of necrotizing fasciitis, impetigo, strep throat, scarlet fever, pharyngitis, and can often cause toxic shock. They are hemolytic, which means that they often grow when they're cultured on blood agar plates. And you can see there on that slide that the yellow is where the bacteria is growing and it has eaten up all of the blood agar around it. Next up is group B, Streptococcus agalactiae. These are typically diplococci, so you'll just see them in those groups of two. This species is the most risk for neonatal infection, UTI, sepsis. So a birthing person may not have any signs or symptoms that they have group B strep, but it can be really dangerous for an infant during birth. And so GBS screenings typically start in the 36th week of pregnancy. And if the birthing person does have group B strep, they're usually given antibiotics about 4 hours before the baby is born. Next is Streptococcus pneumoniae. This species is very resistant to many antibiotics. It's also Diplococci. It has an antigen capsule of polysaccharides around it that typically repel a gram stain and is pretty noticeable when you are growing and culturing it. And this species causes pneumonia, respiratory, and ear infections. For pathogenic species of Streptococcus, penicillin is usually most effective. Erythromycin can be given to people who are allergic to penicillin. It's usually a 10-day round of antibiotics, and you want to make sure and take the full 10 days. The ones that stick around the longest are typically the most resilient, so you want to make sure you kill all of those bacteria so they don't come back stronger later or develop into something more harmful. Streptococcus gets a bad rap, but I wanted to end on a good note. So Streptococcus thermophilus produces formic acid, ferments lactose. It has a positive influence on our gastrointestinal health and is part of a lot of starter cultures. It's non-pathogenic and used in yogurt and cheese production. So typically we think of Streptococcus as being pathogenic, but there are plenty of species of Streptococcus that are non-pathogenic. Thank you so much.
Nocardia, the Great Masquerader. I don't know what's going on. I'm not feeling so well. I wonder if it's my COPD acting up. I'm having a real hard time breathing today. Bob, you really don't look well. We should probably go to the emergency room. Doctor, what do you think is going on with this patient? I have to say I'm really stumped on this one. He's got known COPD treated with long-term steroids. I originally thought it was a simple COPD exacerbation, but he isn't responding to treatment for that. He's also got a fever, elevated C-reactive protein, elevated white blood cell counts, and has started coughing up blood. I worry we may have a case of tuberculosis on our hands. We'll have to do some cultures. Wow, what a great opportunity to be in here. This lung tissue is quite the upgrade. My name is Nocardia asteroidus, and I'm just one of a little over 100 species of bacteria in the Nocardia genus. About half of us, myself included, are pathogenic, although I'm proud to say I hold the title for the most pathogenic. And since I have your attention, let me tell you a little bit about myself. I'm in a class called Actinobacteria, which means I'm a high GC content gram-positive bacteria. High GC means that more than 50% of my DNA base pairs are guanine and cytosine. I'm a very common bacteria and you can find me all over the world. Most often I'm found in soil where I help decay plant matter, although you might sometimes find me in fresh and salt water. I'm a chemoorganotroph, so I do need to get my energy from other sources, which is usually the plants I help to decay. But every once in a while, I get into the air with dust particles. If the right type of person breathes me in, I get to upgrade to the human body. I can also sometimes sneak into the human body through openings in the skin. I'm aerobic bacteria, meaning I need oxygen for energy production and utilize oxidative metabolism. Because I use oxygen for energy production, I'm catalase positive. Catalase is my enzyme buddy that can give oxygen some extra electrons so oxygen can leave my own electrons alone. It's good to hear the doctors aren't on to me yet. Hopefully I can evade them longer. I have a lot of cousins in the Actinobacteria order like Mycobacterium, Corneobacterium, Gordona, Zuckermorel, so hopefully this will continue to throw them off my trail. I also have these cool filament type structures which sometimes develop into clusters called mycelia or develop aerial filaments. Usually we're kind of a whitish creams or orangey color. As I get older, these filaments can change how they appear, breaking them to appear coccyx or rod-shaped. Because of these filaments, people often mistake me for a streptomyces or even a fungus, thank goodness. This is why I've been called the Great Masquerader. I think I should be okay. Hi, Bob. Well, so far we haven't been able to figure out what's going on, so we need to do some more tests. We'll do some more chest x-rays and also take some samples of your blood and respiratory tract to culture them and see what we find. Uh-oh, not culturing. They're being real serious about trying to find me now. I think I should still be able to hide away. If they weren't fooled into thinking I'm a mycobacterium or fungus, thankfully I grow pretty slowly, so either often labs don't let a culture grow long enough to find me. And since they take samples from non-sterile sources, there are so many other bacteria here with me that grow faster, so I'll be pretty well hidden. Can you come look at this for a minute? I think I may have found nocardia in the sample, although I don't know if I believe what I'm seeing. I've never seen it before. Wow, I think you might be right. I've never seen an actual case before, only read about it in medical school. It's so rare. Do you think we just found this by chance? I read it can be found in soil and dust. Maybe there was an accidental contamination of the sample. We're going to want to confirm with mass spectrometry to be sure it's nocardia, but if that's truly what it is, that's likely the cause of his disease. Nocardia is not a bacteria naturally found in the human flora, so we learned that if you see it in a culture, you really need to investigate it further. While we're waiting for the confirmation that this is nocardia, I'll go ahead and start him on some broad spectrum antibiotics just to be sure. Darn it, I've been discovered. Okay, I don't think hope is lost. I've got a few other tricks up my filaments to keep me safe. First of all, I'm a lysozyme resistant. Lysozyme is an enzyme that breaks down the cell walls of bacteria, so I should be pretty safe from that. I'm also a partial acid-fast bacteria, meaning I have mycolic acid in my cell walls. This waxy substance makes my cell walls even tougher. It's also going to be hard to find the right antibiotic to take me out. A lot of us nocardia are drug resistant, and they have to do so much special testing to figure out which antibiotic is going to work. No way will they take the time to do that. Okay, Bob, we got the confirmation back from testing, and it's nocardosis that we're dealing with, so we're going to have to be aggressive with treating you for this. Nocar what? I've never heard of this before. Where could I have even caught this? Are you a gardener or do a lot of yard work? I sure am. I love to garden. It's my favorite hobby. You should see my tomato plants this summer. They are the best they've ever been. If I get out of here okay, I'll have to bring some by for you. So this bacteria, the one we found on your culture, is actually a very common bacteria to find in the environment and soil especially. It's likely that when you're gardening, you breathe in some dust from the dirt with this bacteria in it. That's unbelievable. Is my wife going to be okay? I'm worried she's going to catch this too. She should be okay. It's not contagious human to human, and when people become sick from this bacteria, it is usually when you have some other condition that affects your immune system. In your case, your COPD makes you more susceptible, as do the steroids you've been taking for your COPD. Luckily, your wife doesn't have these immune issues, so she should be okay. And thankfully, we caught it before it became worse. Nocardosis can spread to other organs, including the brain and central nervous system, skin, and soft tissue. So what do we do now? Well, you're already on some antibiotics, but we will need to figure out which ones this infection will respond to. So we're going to have to do some testing. Once we figure out which antibiotic is going to work, you'll have to be on it for many months, maybe a year or more. This bacteria is pretty tough, and it takes a long time to treat. I guess I didn't get away with it after all.
Mycobacterium tuberculosis. Discovered in 1882 by Robert Koch, it is a high G and C gram-positive bacterium, obligate aerobe, slow-growing, and has a round shape. Its mycolic acid cell wall is linked to a arabinogalactan and a thin layer of peptidoglycan. The cell wall lets it resist desiccation, lyses for macrophages, and provides protection against detergents. Several antibiotics and makes it difficult to stain. An acid-fast stain is used to stain this bacterium. The reason they are slow to grow is due to the synthesis of mycolic acid. Mycobacterium tuberculosis is non-spore-forming, chemoorganotrophic, and not a non-motile bacterium. Its length is between 2 to 4 micrometers and its width is between 0.2 and 0.5 micrometers. It has a single circular chromosome, is considered metabolically flexible with the ability to adapt in different environments once it's inside a host. It is flexible in the ability to adjust to different pH levels when moved to new environments within the host. pH ranges between 4.5 to 6.4. Mycobacterium tuberculosis falls under the domain Bacteria, phylum in class Actinobacteria, order Actinomycetales, family Mycobacteriaceae, genus Mycobacterium, species Tuberculosis. Closer relatives are those in the Mycobacterium tuberculosis complex, such as Mycobacterium bovis, Mycobacterium canneti, and Mycobacterium africanum. The MTC is a group of microorganisms that are placed in a complex due to its 99% similarity in regards to its nucleotide level and identical 16S RNA sequences. In ideal aerobic conditions, it will utilize glucose and triglycerides as their primary carbon source during early replication. Once engulfed by a macrophage in the absence of glucose, it will switch to lipids. The genome consists of 9% of it dedicated to lipid metabolism. Inside a macrophage, to preserve carbons by not going through the Krebs cycle, It can utilize the glyoxylation cycle to metabolize fatty acids as the primary carbon source. When stressed, it can protect its iron reserves by the expression of a regulatory RNA. Carbon, sulfur, and nitrogen metabolism distinguishes pathogenic Mycobacterium tuberculosis. It has the ability to break down multiple carbon sources at the same time. Individuals infected by Mycobacterium tuberculosis have shown a greater number of anaerobic microorganisms in their fecal microbiome, such as anaerostypes, BLASHA, correlated to the inflammatory response by the host as a result to TB. Relapsed TB individuals have an increase in actinobacteria, proteobacteria, and a decrease in Bacteroides in their fecal microbiome. Healthy human lungs have four common phyla: Firmicutes, Bacteroides, Actinobacteria, and Proteobacteria. This biosis increases a person's susceptibility to tuberculosis, reactivation of TB, and influences the efficacy of TB treatment. Tuberculosis treatment, such as the use of antibiotics, can result in the dysbiosis and affect the human immune system. Mycobacterium tuberculosis in the environment is not something that is actively being investigated. It may be due to challenges in tracing the microorganism and the fact that it is an obligate human pathogen. Past efforts to link transmission resulted in only being linked to approximately 10 to 30% of cases. However, there are two studies that have been done, one in 2014, where soil was inoculated with Mycobacterium tuberculosis and the bacterium was able to survive for 12 months. It was then added to food and given to mice for 2 months, after which they all developed granulomas. In another study in 2015, 1,500 randomly collections of soil and water in 3 counties in Iran resulted in the isolation of Mycobacterium tuberculosis in 1% of the soil samples and 10% of the water samples. It was then stored for 9 months to see if it would survive, and it did. Tuberculosis caused by Mycobacterium tuberculosis is a worldwide health problem, ranked the 13th leading cause of death worldwide. In 2021, 1.6 million died of tuberculosis. HIV can increase mortality rates in TB patients. Mycobacterium and HIV have a synergistic relationship. Mycobacterium tuberculosis can help HIV replicate, and HIV weakens the immune system, which in turn allows TB to thrive. Tuberculosis infection occurs through an airborne droplet nuclei that is 1 to 10 micrometers in diameter from an infected person when they cough, speak, or sneeze. The droplet goes to the alveoli in the lungs. Macrophages in the alveoli ingest the bacteria but are unable to digest it. Bacteria then replicate within the macrophages, slowly killing it. When the macrophages die, the bacteria are released and the cycle starts again. However, infected macrophages present antigens to T cells that attract more macrophages. The macrophages surround the bacteria and form a tubercle for about 2 to 3 months. During this time, collagen fibers are added to further close off the infection. Cells in the middle of the tubercle die, releasing Mycobacterium tuberculosis. However, it is released as a caseous necrosis that remains in the tubercle. Unless the tubercle ruptures, it will not cause secondary tuberculosis. If a person is exposed, they may develop acquired immunity within 2 to 10 weeks. Individuals are considered to have the infection but not the disease in this case. Over time, the risk decreases of developing tuberculosis. However, it is a risk that never goes away. It stays throughout the person's lifetime. Only 5% of people who have the infection will develop active tuberculosis, which mainly occurs in the first 2 years after the infection. Thank you.
Hello, everyone. Today I'll be presenting my infographic on Escherichia coli, or E. coli, a fascinating bacterium with both beneficial and harmful traits. E. coli is often associated with food safety concerns, but as I'll show, there's a lot more to it, with some strains being essential for human health and scientific research. E. coli belongs to the Enterobacteriaceae family, and it is classified as a gram-negative rod-shaped bacterium. In terms of size and its morphology, it's about 1 to 2 micrometers in length and 0.5 micrometers in diameter. This rod shape, along with its gram-negative classification, plays a role in how it interacts with the environment and responds to various treatments. One of E. coli's defining traits is its metabolic versatility. As a facultative anaerobe, it can switch between aerobic respiration as well as fermentation based on oxygen availability. This allows it to thrive in diverse conditions, and it primarily uses glucose for energy, but can metabolize other substrates like lactose and amino acids, making it adaptable to many different environments. Interestingly, E. coli is a normal resident of the human gut and plays a beneficial role in digestion as well as nutrient absorption. It also synthesizes vitamins like vitamin K and certain B vitamins, which are essential for our overall health. So by living in symbiosis with us, E. coli supports our microbiome, helping maintain balance and preventing the overgrowth of harmful bacteria. However, not all E. coli strains are friendly or beneficial to us. Certain pathogenic strains, such as E. coli O157H7, can cause some serious foodborne illnesses. These strains possess certain virulence factors like toxins, adhesion mechanisms, and immune evasion strategies, which enables them to infect hosts and cause diseases ranging from gastroenteritis to hemolytic uremic syndrome, which are severe complications. E. coli is widespread in nature and can be found in water, soil, and the intestines of warm-blooded animals. Pathogenic strains can spread through contaminated food, contaminated water, or direct contact, making E. coli a significant public health focus when it comes to those certain pathogenic strains. However, it's worth noting that the majority of the E. coli strains are harmless or even beneficial, such as the ones that live in symbiosis with us, to their beneficial to the hosts. Beyond its role in health, E. coli is crucial in scientific research. It's a model organism in molecular biology used for cloning, certain gene expression studies, and more. Its genetic diversity allows researchers to study variations and adaptations, providing insights into microbial evolution and resistance mechanisms. Certain E. coli strains are engineered for bioremediation to clean up pollutants in the environment, highlighting the bacterium's versatility and importance in biotechnology. However, antibiotic resistance is a growing concern as some E. coli strains have developed resistance to multiple antibiotics, which complicates treatment options. E. coli was first identified by Theodor Escherich, after whom it was named in 1885, and has since become one of the most studied microbes that we know of. In the human gut, it's a member of our normal flora where it maintains microbiome balance and supports digestive health, as I mentioned previously. Its presence is usually harmless, helping to keep harmful bacteria at bay. Another notable trait is its ability to ferment lactose, producing acid and gas. This characteristic is used in labs for identification through certain testing protocols, similar to how we do in our labs, in our microbiology labs here. So on nutrient agar, E. coli colonies will appear as pale, round, and moist, which also helps scientists identify it to a certain extent in different mixed cultures. So wrapping up, in conclusion, E. coli is a complex bacterium with a range of roles, from aiding in human health and scientific research to presenting challenges as a harmful pathogen to humans. Its versatility and adaptability make it one of the most impactful microbes, both in our daily lives and in the scientific community. Thank you for listening. Have a nice day.
Hello, my name is Caitlin, and today I'm going to be talking about cyanobacteria. These ancient photosynthetic prokaryotes have shaped the history of life on Earth, starting from their role in oxygenating the planet billions of years ago. Cyanobacteria are key players in ecosystems, contributing to nutrient cycling, oxygen production, and even biotechnology. They are also known for their harmful algal blooms, which makes them a double-edged sword in environmental systems. Cyanobacteria are classified as prokaryotes, meaning that they lack a nucleus, which distinguishes them from algae despite their historical name, blue-green algae. Morphologically, they display a wide range of forms, including unicellular, filamentous, and colonial structures, with shapes ranging from spherical to spiral. Their pigments, such as chlorophyll A and phycobiliproteins, give them their characteristic blue-green color and allow them to perform oxygenic photosynthesis. Cyanobacteria first appeared around 3.5 billion years ago, with fossil evidence in structures like stromatolites. Their oxygenic photosynthesis was a groundbreaking innovation, driving the Great Oxidation event about 2.4 billion years ago in transforming Earth's atmosphere. Today, cyanobacteria thrive in diverse environments, freshwater, marine systems, deserts, and even polar regions, thanks to their metabolic versatility. While they're primarily autotrophic, some species can use organic compounds in the absence of light. These microorganisms also have modern applications. They are being researched for biofuel production, pharmaceutical development, and wastewater treatments, showing their potential to address global challenges, which I think is very interesting. So cyanobacteria play vital roles in ecosystems through their interactions with other microorganisms, organisms, and the environment. A notable example is nitrogen fixation. So cyanobacteria like Anabaena, which I crocheted an Anabaena cyanobacteria for my project. And it's this little guy here. He has a smiley face. But this is the cyanobacteria, and these big guys right here, these are heterocysts, which are actually what aid the process of the nitrogen fixing here. So these Anabaena and Nostoc cyanobacteria, they convert atmospheric nitrogen into bioavailable forms, benefiting plants and enriching soil fertility. So in rice paddies, this process reduces the need for chemical fertilizers. And cyanobacteria also form symbiotic relationships, such as with fungi, enabling them to survive in extreme conditions. Now, cyanobacteria are also known for their harmful algal blooms. So these occur when nutrient-rich waters, often from agricultural runoff, create the perfect conditions for cyanobacteria to proliferate. So these blooms disrupt ecosystems by depleting oxygen, killing aquatic life, and releasing toxins harmful to humans and animals. These are further exacerbated by climate change and warming water temperatures. This duality, being both essential and potentially harmful, highlights the cyanobacteria's complex role in ecosystems. So one of those cyanobacteria that can cause those harmful algal blooms is called Microcystis aeruginosa. And this is a well-known cyanobacterium. So this unicellular species often forms colonies surrounded by a mucilage sheath, which provides protection and buoyancy in aquatic environments. And this is closely related to other bloom-forming cyanobacteria like Anabaena and Plankthrix. One of the most concerning aspects of this is its ability to produce microcystins, which are potent toxins that can contaminate drinking water and harm aquatic life. These toxins protect organisms from oxidative stress and predators, giving it a competitive edge in eutrophic environments. And factors like high nutrient levels and warm temperatures often trigger blooms, and they create ecological and public health challenges. So in conclusion, cyanobacteria are extraordinary microorganisms with a dual nature. They've played a pivotal role in Earth's history by oxygenating the atmosphere and continuing to shape ecosystems through nutrient cycling and primary production. However, their ability to form harmful algal blooms underscores the importance of managing their ecological impacts. With ongoing research, cyanobacteria also hold the promise of addressing global challenges in sustainability, renewable energy, and environmental conservation. Okay, thanks for listening.
My chosen organism is Clostridium botulinum. It is a gram-positive spore-forming anaerobic bacterium that produces botulinum neurotoxins, the most potent toxin known to humans. It appears as single cells, in pairs, or in chains. And like mentioned in class, as a spore-forming organism, it produces oval, highly resistant endospores that ensure survival under extreme environmental conditions. And these spore-forming capabilities allow it to persist in diverse environments. Clostridium botulinum is part of the genus Clostridium, a group of gram-positive spore-forming anaerobic bacterium. Its phylogeny is complex, and it reveals 4 distinct lineages based on biochemical traits and 16S ribosomal RNA sequencing. These groups vary in enzyme activity, growth temperature ranges, and associated toxin types. Historically, all neurotoxin-producing Clostridia were classified as Clostridium botulinum. However, recent studies have shown that closely related species, such as the ones listed over here, can also produce those neurotoxins, suggesting horizontal gene transfer. So, botulinum neurotoxins are classified into 7 serotypes, A to G, and essentially what this image is showing is how those neurotoxins block nerve to muscle signaling, and this causes botulinum, and this causes botulism, and it is a rare, severe neuroparalytic illness, and symptoms include muscle paralysis, respiratory failure, abdominal pain, nausea, vertigo, muscle weakness, trouble breathing, and without proper treatment, it can lead to death. Clostridium botulinum thrives in anaerobic conditions, like mentioned before. It shares habitats like soil, sea sediments, and decomposing organic matter with other microbes, and it influences surrounding populations by producing neurotoxins that exploit hosts and potentially alter nutrient dynamics. In human intestines, it germinates alongside bacteria, and it impacts microbial communities. The bacterium interacts significantly in food microbiology, where it thrives in poorly sterilized anaerobic conditions and spoiled food. And in humans, it causes botulism through foodborne, wound, or intestinal infections. And its neurotoxins impact humans and animals alike, with serotypes C and D affecting specifically animals. And though it's rare today, just in the US alone, they are seeing 150 cases annually, so botulism remains a significant food safety concern. Again, because it thrives in low oxygen environments, improperly processed home canned goods and low fat acid foods are at risk of getting that infection, and the bacterium spores resist boiling temperature, making pressure necessary for canning for safe food preparation. So just to recap, Clostridium botulinum is a gram-positive spore-forming anaerobic bacterium that produces botulinum neurotoxins. These toxins, classified into 7 serotypes A to G, cause botulism. And botulism is a severe neuroparalytic illness with symptoms like muscle paralysis and respiratory failure. The bacterium thrives in anaerobic, so low oxygen environments like soil, sea sediments, and improperly processed foods, particularly in low acid home canned goods. Its spores, highly resistant to extreme conditions, germinate under favorable conditions, impacting humans, animals, and microbial ecosystems. I hope you enjoyed the presentation. Thank you.
In 2004, a groundbreaking discovery in the Netherlands introduced the world to Akkermansia muciniphila, a species of the Akkermansia genus. This bacterium lives within us, quietly revolutionizing our understanding of the human microbiome. Since then, this microscopic ally has emerged as a critical player in gut health and beyond. Akkermansia muciniphila is an anaerobic, gram-negative, rod-shaped bacterium thriving in the mucosal layer of the human gut. Representing about 3% of our gut microbiota, it specializes in breaking down mucin, the glycoprotein that forms a protective lining in our intestines. It is a member of the phylum Verrucomicrobia, with many relatives who share ecological niches but do not exist in the gut microbiome. By breaking mucin into simpler sugars and producing beneficial compounds like acetate and propionate, Akkermansia plays a pivotal role in maintaining the gut barrier. These short-chain fatty acids support gut health, reduce inflammation, and help prevent leaky gut syndrome, a condition linked to autoimmune diseases, mood disorders, and skin conditions. The presence of Akkermansia is often associated with metabolic health. Research shows that higher levels of this bacterium correlate with lower risks of obesity, type 2 diabetes, and cardiovascular disease. Its ability to regulate gut permeability and modulate immune responses makes it a promising candidate for future probiotic therapies. But how does it work, you may wonder? Akkermansia doesn't just live in the gut, it actively shapes the environment. By degrading mucin, it releases nutrients that other beneficial bacteria can use, indirectly suppressing harmful pathogens and fostering a balanced microbiome. Recent studies have shown that dietary supplementation with Akkermansia can improve glucose metabolism, reduce gut inflammation, and strengthen the gut barrier. However, not all individuals may benefit equally. Conditions like inflammatory bowel disease and polycystic ovarian syndrome, where the gut lining may already be extremely compromised, require careful consideration, as supplementation could lead to complications. Interestingly, while Akkermansia is a marker of metabolic health, an overabundance in the gut has been indicated as a marker for neurological disorders like Parkinson's disease and multiple sclerosis. This dual nature underscores the need for personalized approaches when exploring its therapeutic potential. The future of Akkermansia research is bright. With the identification of 8 new strains in 2022, the scientific community is poised to unlock even more ways this bacterium can contribute to human health. From targeted therapies to precision probiotics, Akkermansia is set to play a central role in the next generation of gut health solutions. Understanding Akkermansia muciniphila means understanding ourselves. Small but mighty, it's a reminder that the key to health often lies in the tiniest places. Thank you.
Today I'm talking about Yersinia pestis, the bacterium responsible for one of history's deadliest pandemics. Known for causing the Black Death in the 14th century, this gram-negative bacterium is still present today and remains a zoonotic threat. Yersinia pestis is a gram-negative, rod-shaped bacterium in the family Enterobacteriaceae. Its outer membrane protein, called AIL, helps it to attach to host cells and avoid destruction by the immune system. These features allow it to infect mammals, including humans, and cause severe disease. Yersinia pestis shares a close evolutionary relationship with Yersinia pseudotuberculosis, from which it diverged around 15 to 2,000 years ago. While Y pseudotuberculosis primarily causes mild gastrointestinal illness, Y pestis adapted to survive in both mammals and fleas, developing a much greater virulence. This evolutionary shift helped it spread rapidly and cause pandemics. Yersinia pestis is a facultative anaerobe, meaning it can grow with or without oxygen. This metabolic flexibility allows it to survive in diverse environments, such as flea guts or mammalian tissues. It also uses a specialized iron acquisition system to thrive in iron-poor environments, like the host's bloodstream. This system is critical for its survival and pathogenicity. The life of Y. pestis involves flea vectors and rodent hosts. Inside fleas, it forms biofilms in the digestive tract, which block the flea's ability to feed. This blockage causes the fleas to bite aggressively, transmitting the bacterium to mammals. This mechanism ensures that the bacterium's survival and spread through new hosts. Y. pestis affects ecosystems by occasionally reducing rodent populations, which can disrupt predator-prey dynamics. For instance, outbreaks in prairie dog, Cynomys lodovicianus colonies in the Badlands of South Dakota highlight the need for flea control and rodent monitoring to prevent larger epidemics. In humans, Y. pestis causes 3 types of plague. Bubonic plague, this causes swollen lymph nodes or buboes and has a mortality rate of 30 to 75% if left untreated. Pneumonic plague infects the lungs, and septicemic is very rare but is a blood infection and is almost always fatal without treatment. Prompt medical intervention, such as antibiotics, is essential to save lives. Y. pestis has several virulence factors that make it highly effective at causing disease. The EIL protein enables it to resist immune defenses, while the plasminogen activator enzyme breaks down host tissues, allowing the bacterium to spread rapidly. These features help Y. pestis invade, evade, and infect with deadly efficiency. Historically, Yersinia pestis caused devastating pandemics, such as the Black Death, which killed an estimated 25 million people in Europe. Today, it remains endemic in certain areas, with occasional outbreaks. This persistence reminds us of the need for ongoing public health measures to monitor and control this pathogen. In conclusion, Yersinia pestis, a pathogen of historical and modern importance. Its adaptability, ability to evade the immune system, and survival strategies make it a remarkable microbe. And by studying Y. pestis, we continue to learn about combating infectious disease and improving public health. I made an art project for our assignment. I researched classic ornament and historical pictures and images of the plague, and put together a collage of monoprints using an acid transfer or an acetone transfer pen. And it was really fun, even if it was such a sad topic to work on. It was really great to make something. And I wanted to do it. I did the printing on the back of sewing pattern tissue paper because I wanted the conceptual idea that this touched so many bodies.
Hey guys, my name is Sanai, and I decided to research on a bacteria that is called Rickettsia. Rickettsia is a bacteria that is found in ticks, lice, fleas, mites, and mammals. This is a gram-negative bacteria. It is an obligate intracellular parasite that synthesizes only a small amount of peptidoglycan. It is non-motile and anaerobic. It lives in the cytosol of their host cells, since outside of the host cells it's unstable and dies quickly. This is an extremely small bacteria, as it only measures 0.3 micrometers times 1.0 micrometer. It is typically a bacilli, rod-shaped. These are usually defined by 2 groups, spotted fever group and a typhus group. Rickettsia is a class alpha proteobacteria of the bacteriophyllum Proteobacteria. This is based on the sequence of nucleotides in their rRNA molecules. There are 4 genera: Rickettsia, Aurantia, Ericia, and Anaplasma, which are the main rickettsias that cause disease to humans. Their genes encode 16S RNA. Two of the specific species from Rickettsia are R. corori caspia, which is an Astrakhan spotted fever, and R. africae, which is an African tick bite fever. Rickettsia requires a vector of transmission from host to host. Rickettsial species are transmitted by a different interpoled vector, and when inside of the host cell, they secrete an enzyme that digests the membrane of the endocytic vesicle, which will release a bacteria into the host cell's cytosol. They possess an outer membrane of lipopolysaccharide with endotoxin activity. They reproduce by binary fission. Rickettsia also spread via the bloodstream to infect the endothelium and sometimes the vascular smooth muscle cells. Rickettsia also are able to synthesize ATP via metabolism of glutamate. There are 3 main types of rickettsia. The first one I'll be discussing is Rickettsia prowazekii. This causes epidemic typhus since it's vectored by the human body loci. This usually occurs when scratching inoculates bacterium into the skin. Humans are the primary host, and they usually fills the host cells until the cell breaks open. This usually occurs in unsanitary conditions, fever, body lice. Some things that it can manifest is high fever, mental and physical depression, plus rash that can last up to 2 weeks. Brozinger is a recurrent disease that is more mild. It is a more mild type of rickettsia. The next type of rickettsia is Rickettsia typhi, which is a murine typhus. It is a major reservoir for bacterium in rodents. It is a rat and cat flea vector that transmit an animal host to humans. Following the 12 days after the bite of an infected flea, you can see fever, severe headache, chills, muscle pain, and nausea occur. This usually lasts up to 3 weeks, and if left untreated, is rarely fatal. There is no vaccination available for this type of disease or bacteria. The main and most common type of severe rickettsia is Rickettsia rickettsii, which is the most severe as mentioned. This usually causes the Rocky Mountain spotted fever. And this is especially true for people with the glucose 6-phosphate dehydrogenase deficiency. It is infected by the Dermacentor variabilis tick. The treatment choice consists of doxycycline, which can be used in people of all ages, including young children. This bacteria spreads via the bloodstream and damages the lungs, brain, and other organs. Mortality rates are as high as 20 to 30%. The image that I have included here talks about the merging and remerging rickettsia cycle, which starts with an uninfected tick, which will then attach to an infected rodent, and then potentially infecting the tick as well, which will then attack an uninfected rodent, and it would just become a cycle where they would lay their eggs and then proceed to infect an adult, which will then lead to an infected adult. And it's just a cycle that keeps continuing and going on and on. Thank you for listening to my presentation.
Bifidobacteria, a beneficial microbe. Bifidobacteria are a genus of gram-positive, anaerobic, non-motile, and non-sporulating bacteria. They have about 63 different species, and some of them are polymorphic. The most commonly found species in humans are B. adolescentis, which is the only species of bifidobacteria that is intrinsically Y-shaped, B. bifidum, B. longum, and B. infantis. Where can these bacteria be found? They can be found in the human gastrointestinal tract, primarily in the large intestine. They can be found in fermented foods such as yogurt and kimchi, and they can also be found within insects. Bifidobacteria make up a significant portion of the gut microbiota. They are the predominant phylogenetic group in early human life, and they play a huge role in the breakdown of carbohydrates such as fiber and human breast milk from our diet. Bifidobacteria use a unique metabolic pathway in their fermentation process. They use what is called the bifid shunt, or fructose 6-phosphate shunt. Bifidobacteria use this special pathway to convert sugars into intermediates, which are then processed further into short-chain fatty acids. Bifidobacteria can ferment a variety of plant-derived carbohydrates, including pectin and oligosaccharides found in fruits, vegetables, and legumes, though the specific carbohydrates they can metabolize depend on the strain. The short-chain fatty acids that they produce are high in caloric content and are absorbed by cells in the large intestine, where they are metabolized and used as an energy source. These short-chain fatty acids not only provide energy, but also help maintain gut health by supporting the intestinal lining, enhancing water and sodium absorption, and promoting the healing of gut tissues. The bifidobacterial shunt yields 2.5 ATP molecules. Fun fact, bifidobacteria can partake in cross-feeding. Certain strains of bifidobacteria can break down large and complex molecules and feed them to other strains. This symbiotic relationship makes it possible for both species to survive in their environment. In addition to their help with digestion, bifidobacteria also play a role in the prevention and treatment of certain diseases. Research has found that a B. longum and B. breve strain provide protection of DNA from induced damage from carcinogens. Bifidobacteria has also been found to reduce and treat various GI issues such as inflammatory bowel disease, necrotizing enterocolitis, and diarrhea. In conclusion, Bifidobacterium species are essential members of the human gastrointestinal microbiota, playing a crucial role in promoting gut health, especially during early life. Their ability to metabolize a wide range of complex carbohydrates highlights their adaptability and importance in maintaining a balanced gut microbiome. Through their diverse genetic profiles and enzymatic capabilities, bifidobacteria contribute significantly to human health, making them a key focus of research in microbiology and probiotics.
Hi, I'm Neisseria gonorrhoeae. I'm a gram-negative Diplococcus obligate anaerobe bacteria. You've probably heard of me from your sex ed class, friends, or maybe even your partner. I'm an obligate human pathogen, which means I love people. I come from a big family, but only one of my siblings shares the same love for people that I do. Her name is Neisseria meningitis. She's the leading cause of bacterial meningitis. She's a little bit less serious about her love for people than I am, but she does seem to cause a bit more harm to her host than I do too. Also, people seem to find out about her faster than they do me. My other siblings in my genus are similar to me, but have other passions. We all like being in wet environments that are about 98.6 degrees Fahrenheit. We all enjoy being on animals' mucosal surfaces, and we are built to thrive in those environments. Before we get into my wants and loves, let's talk about my needs. I'm an obligate aerobe, which means I need access to oxygen to survive and thrive. For nutrition, I'm a little needy. In a lab, I need a special agar to grow. I do best on blood agar and chocolate agar. Yum. In the most basic terms, I need glucose, glutamine, thiamine, phosphate, iron, and carbon dioxide in order to thrive and grow. Now for my favorite topic, people. They've been obsessed with me for as long as I have been obsessed with them. A bunch of famous people have even talked about me, including Hippocrates, who I think is a pretty big deal. I'm even mentioned in the most printed book in history, the Bible. Each year worldwide, more than 80 million people find out that I am with them, and The WHO even recently named me a superbug due to my obsession with humans and adaptability to their treatments. I usually find a new person to be with through the sexual contact of two people. You could say I love love. Once I find a new host, I bind to their epithelium in their throat, vagina, or rectum. When I first get there, I try to hide my best from the host body. Even though I love them, the immune system of my hosts don't really love me back. Depending on which zone I end up in, I use different techniques to hide from the body. In the cervix, for example, I bind to factor H, which the host body identifies as itself so it doesn't attack me. Once I get into a host, I use a few different proteins in my outer membrane called TDTSs that work to hoard some nutrients away from other organisms in the area, to both suppress the other organisms and pathogens and keep some resources for myself in case I need it later. Since I'm exclusive to humans, I had to learn how to adapt to avoid their rude medical advances and efforts to get rid of me. I use biofilms and horizontal gene transfer using my pili to share information with my peers and learn from their previous experiences with these things called antibiotics. Luckily, every time someone survives one of those series, we can share insider knowledge and become stronger, and build resistance to these horrible medications. Despite my efforts, scientists are still trying to figure out how to get rid of me. Once I make my new home in my human host, I'm contagious about a week after my first contact with them. Despite that, my host will still test negative for about a week after I'm contagious. That's still not long enough for me, but luckily it takes a lot longer for most people to find out that I'm even there. In female hosts, I typically fly under the radar. I usually do not cause noticeable symptoms in female hosts for a long time. Most find out about me through random routine screening at a doctor's office. I do cause some symptoms in male hosts, but it's usually just some discharge. Even though I love them so much, I can see why they don't like me. If I go undetected for long enough, I can cause some harm to my hosts. 1 in 5 females who do not receive treatment for me will get pelvic inflammatory disease, which can cause scarring and have long-term fertility effects, and requires a long course of medication to cure. Additionally, if I'm in a pregnant person, I can cause harm to the baby. I cause neonatal blindness and scarring and ulcers on the cornea of the infant. Thank you so much for taking the time to learn all about me.
Clostridium difficile, aka C. diff, is the main culprit of a healthcare-associated infection that causes severe diarrhea. C. diff was discovered by Louis Pasteur in the 1860s and was first isolated from the intestinal flora of newborns in 1935. It was originally named Bacillus difficile because it was difficult to culture and study. C. diff is a gram-positive, obligate anaerobe, bacillus-shaped bacterium and has a vegetative form and spore form. Because it's an obligate anaerobe, it cannot survive in the environment for very long when it's in its vegetative state. When it's in its spore form, it is metabolically inactive and can withstand oxidative stress, extreme temperatures, desiccation, and acidic environments. The C. diff spores in the stool of those who are infected can live in the environment for extended periods of time, as they can survive harsh conditions, including the surfaces that are sanitized with alcohol. C. diff is part of the phylum Firmicutes and has many relatives, including Clostridium perfringens, which can cause food poisoning and diarrhea, and Clostridium tetani, which can be found in soil and cause spastic paralysis. When C. diff enters its host, it navigates through the acidic environment of the stomach and then travels to the small intestine where it germinates. For germination to occur, germinants and the proper nutrients must be present in the environment. Bile acids, along with glycine and other amino acids, trigger the germination of C. diff spores. Glycine and the other amino acids act as co-germinants. The spores then move into the anaerobic environment of the large intestine where they multiply and colonize. They then attach to the epithelium of the colon and secrete toxins that cause the infection. C. diff is found in soil, feces, sewage, and food and is transmitted through the fecal-oral route. CCPA is the main carbon catabolite regulator that also reduces the toxin gene expression. It uses the Stickland reaction, which oxidizes and reduces glycine and proline, which produce ATP and NAD+. When there's a limited supply of proline or proline reductase, alternative pathways are used to regenerate NAD+, which produces butyrate, which then stimulates the synthesis of the toxins of C. diff. Antibiotic use decreases the overall diversity of the intestinal microbiome. Specifically, broad-spectrum antibiotics decrease the amount of bacteroidetes, which allows germination and growth of pathogenic microorganisms such as C. diff. When the gut microbiota changes, the number of metabolites in the intestine are also affected, which alters bacterial fermentation. This decreases the amount of fatty acids while increasing carbohydrates and amino acids, which allows C. diff to use these metabolites for germination, colonization, and growth. Those who are at high risk for contracting C. diff infection include the elderly with multiple comorbidities who frequently undergo urgent surgery. Those who become infected with C. diff tend to be readmitted to the hospital, which provides the pathogen with more opportunities to spread to its next victims.
For this presentation, we're looking at the microorganism Helicobacter pylori. Helicobacter pylori is a gram-negative, non-fermenting, non-sporulating microaerophilic bacteria. It's about 2 to 4 micrometers long with a diameter of about 1/2 to 1 micrometers. It has a helical shape with about 2 to 7 Lophotrichus flagella at the end. That means that all the flagella are used for movement in one direction. It does come in a couple other shapes, but the other shapes are not as useful, just in rods or curved. The spiral shape is the one that's most successful and impactful. Helicobacter pylori is within the bacteria domain, and as you can see on this slide, it shows the phylum, class, order, family, and then the genus and species, which is Helicobacter pylori. This microorganism is a highly successful and prevalent bacteria. I saw a variety of different estimates in terms of the prevalence, but one that I saw is approximately 2/3 of all people in the world are believed to have been infected at some point, with higher prevalence in the developing world. The vast majority of people who have this bacteria are asymptomatic, but those who do have symptoms can develop chronic gastrointestinal symptoms. H. pylori is believed to be linked to approximately 89% of gastric cancers, 5.5% of all cancers worldwide, making it the only bacterium linked to development of any cancer. H. pylori has extensive genetic diversity of strains, which helps it thrive. So within one person, there can be a lot of strains, and between different people. The genus Helicobacter consists of over 20 recognized species, with many awaiting formal recognition. In addition to a lot of diversity within humans, there are related Helicobacter species that have been identified in the stomachs of other wild and domesticated mammals. There are a number of features that allow this bacteria to survive and thrive in stomachs. It's microaerophilic, so within the stomach it only needs a little bit of oxygen, and it contains hydrogenase, which it uses to oxidize hydrogen in the surrounding environment to get its nutritional energy. From that hydrogen, it produces oxidase, catalase, and urease. The urease production is key to its survival because it takes it in, breaks it down, and it creates ammonia and carbon dioxide. That ammonia in the surrounding area increases the pH, making it more basic, less acidic, and creates kind of a window of less acidity right around the organism, which allows it to live. When people have an H. pylori infection, it impacts the entire microbiome in a person's stomach. It can be around 40 to 90% of all the microbiota when it's present. So it reduces the overall diversity. The change in pH impacts the ability of other microorganisms to survive. And then another way it can impact the stomach is that eradication therapies can also further ruin the microbiome. It uses its helical shape and flagella to burrow into the mucus lining of the stomach. So it gets through the mucus and up to the skin layer. Once it's in the epithelial layer, it produces adhesins which allow it to stick there and not get washed away. Once it is up against the stomach wall, it stays there and produces ammonia, which decreases acidity. The ammonia allows it to stay there, but the ammonia also is toxic to the epithelial layer, so it ruins this stomach lining. It creates an immune response, more acids. So basically, it sits there against your stomach wall, and then your stomach acid ruins your stomach lining, creating ulcers. With an H. pylori colonization or infection, people can experience abdominal pain, bad taste in the mouth, nausea, heartburn, belching, and the infection is related to a number of diseases: chronic gastritis, peptic ulcers, gastric adenocarcinoma, gastrointestinal reflux disease, and cancer. H. pylori has been isolated in saliva, gastric mucus, dental plaque, and feces. It's transmissible through oral route or fecal oral route. Right now, there are no vaccines, so hand hygiene, access to clean water are really important. There are a number of different antibiotics that can kill an H. pylori infection or colonization. Other therapies like proton pump inhibitors, bismuth salt, and a number of different foods are recommended. And if you're having all of the symptoms in the previous slide, it might be worth going to a doctor and getting checked out.
Mycobacterium tuberculosis. Discovered in 1882 by Robert Koch, it is a high G and C gram-positive bacterium, obligate aerobe, slow-growing, and has a round shape. Its mycolic acid cell wall is linked to a rabinogalactan and a thin layer of peptidoglycan. The cell wall lets it resist desiccation, lysis for macrophages, and provides protection against detergents. Several antibiotics and makes it difficult to stain. An acid-fast stain is used to stain this bacterium. The reason they are slow to grow is due to the synthesis of mycolic acid. Mycobacterium tuberculosis is non-spore-forming, chemoorganotrophic, and not a non-motile bacterium. Its length is between 2 to 4 micrometers and its width is between 0.2 and 0.5 micrometers. It has a single circular chromosome, is considered metabolically flexible with the ability to adapt in different environments once it's inside a host. It is flexible in the ability to adjust to different pH levels when moved to new environments within the host. pH ranges between 4.5 to 6.4. Mycobacterium tuberculosis falls under the domain Bacteria, phylum and class Actinobacteria, order Actinomycetales, family Mycobacteriaceae, genus Mycobacterium, species Tuberculosis. Closer relatives are those in the Mycobacterium tuberculosis complex, such as Mycobacterium bovis, Mycobacterium canneti, and Mycobacterium africanum. The MTC is a group of microorganisms that are placed in a complex due to its 99% similarity in regards to its nucleotide level and identical 16S RNA sequences. In ideal aerobic conditions, it will utilize glucose and triglycerides as their primary carbon source during early replication. Once engulfed by a macrophage in the absence of glucose, it will switch to lipids. The genome consists of 9% of it dedicated to lipid metabolism. Inside a macrophage to preserve carbons by not going through the Krebs cycle, it can utilize the glyoxylate shunt cycle to metabolize fatty acids as a primary carbon source. When stressed, it can protect its iron reserves by the expression of a regulatory RNA. Carbon, sulfur, and nitrogen metabolism distinguishes pathogenic Mycobacterium tuberculosis. It has the ability to break down multiple carbon sources at the same time. Individuals infected by Mycobacterium tuberculosis have shown a greater number of anaerobic microorganisms in their fecal microbiome, such as anaerostypes, Blasha, correlated to the inflammatory response by the host as a result to TB. Relapsed TB individuals have an increase in actinobacteria, proteobacteria, and a decrease in bacteroides in their fecal microbiome. Healthy human lungs have 4 common phyla: Firmicutes, Bacteroides, Actinobacteria, and Proteobacteria. This biosis increases a person's susceptibility to tuberculosis, reactivation of TB, and influences the efficacy of TB treatment. Tuberculosis treatments, such as the use of antibiotics, can result in the dysbiosis and affect the human immune system. Mycobacterium tuberculosis in the environment is not something that is actively being investigated. It may be due to challenges in tracing the microorganism and the fact that it is an obligate human pathogen. Past efforts to link transmission resulted in only being linked to approximately 10 to 30% of cases. However, there are 2 studies that have been done, one in 2014, where soil was inoculated with Mycobacterium tuberculosis and the bacterium was able to survive for 12 months. It was then added to food and given to mice for 2 months, after which they all developed granulomas. In another study in 2015, 1,500 randomly collections of soil and water in 3 counties in Iran resulted in the isolation of Mycobacterium tuberculosis in 1% of the soil samples and 10% of the water samples. It was then stored for 9 months to see if it would survive, and it did. Tuberculosis caused by Mycobacterium tuberculosis is a worldwide health problem, ranked the 13th leading cause of death worldwide. In 2021, 1.6 million died of tuberculosis. HIV can increase mortality rates in TB patients. Mycobacterium and HIV have a synergistic relationship. Mycobacterium tuberculosis can help HIV replicate, and HIV weakens the immune system, which in turn allows TB to thrive. Tuberculosis infection occurs through an airborne droplet nuclei that is 1 to 10 micrometers in diameter from an infected person when they cough, speak, or sneeze. The droplet goes to the alveoli in the lungs. Macrophages in the alveoli ingest the bacteria but are unable to digest it. Bacteria then replicate within the macrophages, slowly killing it. When the macrophages die, the bacteria are released and the cycle starts again. However, infected macrophages present antigens to T cells that attract more macrophages. The macrophages surround the bacteria and form a tubercle for about 2 to 3 months. During this time, collagen fibers are added to further close off the infection. Cells in the middle of the tubercle die, releasing Mycobacterium tuberculosis. However, it is released as a caseous necrosis that remains in the tubercle. Unless the tubercle ruptures, it will not cause secondary tuberculosis. If a person is exposed, they may develop acquired immunity within 2 to 10 weeks. Individuals are considered to have the infection but not the disease in this case. Over time, the risk decreases of developing tuberculosis. However, it is a risk that never goes away. It stays throughout the person's lifetime. Only 5% of people who have the infection will develop active tuberculosis, which mainly occurs in the first 2 years after the infection. Thank you.